Good afternoon. Thank you so much for joining us, and I imagine people will continue to trickle on in. We're really excited and have some wonderful speakers here and a wonderful patient advocate to help us address some of these interesting and pressing questions in cancer genetics. So the title of this session is The Future of Cancer Genetics is Here, and I'm delighted that I'm joined by three outstanding faculty who are going to tackle different issues when we think about risk assessment and how we both identify those at risk, better categorize what their risk is, and then how that applies when we start to think about how that impacts treatment. So without further ado, we're going to go ahead and start with the first talk. And Dr. Fergus, who is the Zbigniew and Anna M. Scheller Professor for Medical Research and the Chair of the Division of Experimental Pathology and Laboratory Medicine, the Department of Laboratory Medicine and Pathology at Mayo Clinic, is going to lead off our session.
Okay, thank you, Claudine, and thank you to the organizers for giving us the opportunity to present here. I'm going to be talking about saturation genome editing of VUS and BRCA2. Here are my disclosures. Okay, just a quick overview of cancer genetics. Most people will be familiar with this map. Essentially what we're doing is showing the various genes that are known to predispose to breast cancer. Frequency is across the bottom, and the risk is vertically, it's shown on the y-axis. And then we have BRCA1 and 2 at the top, very high risk, roughly common. PALB2 below that, CHEK2 and ATM. These are the 5 genes that most people talk about for breast cancer risk. But then we have some more rare genes, such as RAD51C and D, MAP3K1, ATRIP. These were shown last year in a paper in Nature Genetics to be associated with risk, and then syndromic genes as well. In addition to that, we have SNPs, bottom right-hand corner, all these common allelic variants. We will not really be talking about that today, although I think Dr. Antoniou might cover some of that. Now that we know many of the genes that drive risk, how do we use it clinically? So, of course, I think everybody in the audience will know that there's a lot of genetic testing going on, hereditary cancer genetic testing going on. And there are guidelines in many countries for who qualifies for the testing. In this country, it's NCC-based. And we have guidelines that basically say family history of certain cancers, age of diagnosis under the age of 60. These women would all qualify for testing. Multiple cancers also qualifies you. And then we also have calls from various organizations saying we should test all women with breast cancer. That's a topic for a different day, perhaps. And then the US Preventative Services Task Force really focusing on unaffected individuals and who should be tested there. And again, that's really decided by risk stratifying models. The genes, here are the genes that we, again, think of as being associated with breast cancer risk, color-coded by, on the charts there. These are cumulative risk models, so increasing risk over time. And then what we see is that BRCA1 and 2 are very high, and then some of the moderate risk genes, and then finally the rare genes at the bottom. Population levels of risk shown in that light blue color at the very bottom. So all of these genes increase your risk if a mutation is found. But one of the main problems of genetic testing is the identification of variants of uncertain significance. And we've known about these for a long time, but really haven't made much headway in terms of figuring out which ones are pathogenic and which ones are benign. So there are many thousands of these variants known in all of these predisposition genes. And today I'll be talking mainly about BRCA2, where there are about 5,000 of these different variants now identified. They account for still a fairly high proportion of all new test results that are coming out. What's the problem with a variant of uncertain significance? Well, unfortunately, we have to regard a variant of uncertain significance as essentially no information at all. So the patient cannot benefit from any of the factors shown on the bottom of the screen here, such as additional risk assessment and cascade testing for family members, enhanced MRI and mammography screening, prophylactic surgeries, and then, of course, with the advent of PARP inhibitors, can't really benefit from the application of PARP inhibitors because we really have no clue as to whether the variant is driving sensitivity or not. So that leaves a huge unresolved question. Many thousands of women around the world and in the United States that carry these variants, and we don't know really what to do with them. They have to be basically counseled on the basis of their personal history. So to address this issue, we developed what we call a saturation genome editing screen, which is based on CRISPR-Cas9 technology. And here's a graphic showing you how it works. Essentially, you have the BRCA2 gene at the top with the various structures. You can see the region we focused on in that dotted box, exons 15 through exon 26. This is the DNA binding domain of BRCA2, which is a core functional region of BRCA2. This is where all the known pathogenic missense variants are located. So we think of this as essentially the hotspot, even though it's a third of the size of the entire gene. It's quite a large region. So you can see the structure there. And the way we went about this experiment is to go exon by exon. Each exon has to be its own experiment. We do this in haploid cells. So the idea here is that only one copy of the human genome in these cells. So you only have to target the mutation in one time for it to be present in the genome. Essentially, we're integrating hundreds of thousands of variants all at the same time into the genomes of these cells. So it's essentially endogenous effects that we're looking at. It's not overexpression based. It's under normal regulatory control of expression within the cell. We do know that BRCA2 is required for cell viability. So if you knock out BRCA2, these cells will gradually die over a number of days. So in this situation, we're looking at cell survival shown by the Petri dishes at the bottom of the graphic. Essentially what's happening is that if you have a nasty mutation in there, these cells will gradually die away. If you have a benign or neutral variant, these cells will continue to grow. And so you can wait until day 14, harvest the DNA, sequence it to mass depth, and then you just count the number of times you see each variant. And if it's common, it's still growing and it's probably neutral. If there's hardly any copies of the variant there, it's been dying away and it's probably deleterious or pathogenic, potentially pathogenic. So essentially that's the way we do it. It's all viability screens within cells followed by deep sequencing and then bioinformatics. So these are the results of our BRCA2 analysis of critical functional domain. I'll bring your attention particularly to the orange and to the light blue colors. These are our controls. These are known sensor protein truncating mutations in the orange. Synonymous variants, so no amino acid change. So probably benign, probably neutral variants in the light blue color. And you can see how nicely distributed they are on the x-axis. The x-axis is a computed functional score, which is essentially the log of the day 14 number of copies of your variant divided by the day 5 number of copies of your variant. It's a very simple ratio. But you can see there's a very nice separation of these types of variants. The green color are all the other variants, all the missenses that remain VUS, if you want to think of them as VUS. These are all sitting there, and they're nicely distributed too. And so you can see there's many of the green ones are sitting on top of those orange protein truncating variants, suggesting that these are nasty mutations that are potentially pathogenic. So here's a summary of the results. We did, in this assay, we included all possible single nucleotide changes. So consensus splice are there, protein truncating nonsense variants are there, as well as missense variants. So of the 342 nonsense variants that were in this data set, we essentially, I think you can see, we got 99% of them to function as P-strong. And so if you look at the category on the right, these are 7 categories of pathogenicity that we've assigned based on these function scores. So this is pathogenic, strong, moderate, supporting, and benign, supporting, moderate, and strong, with VUS still in the middle. Essentially, this is based on the ACMG, the American College of Medical Genetics guidelines for classification of variants. They recommend these categories, and we can use likelihood ratios, or probabilities converted to likelihood ratios, to assign variants based on the functional score. We can assign them to these 7 categories of probability. So the pathogenic strongs are really the nasty-looking ones, and all the nonsenses are occurring in that category. If you look at the bottom, nearly all the synonymous variants, so again, no amino acid change, probably a neutral variant, those are all occurring in the benign categories. So that makes sense. These controls are operating very well here. And then in the missense group in the middle, 4,583 of them, you can see that there's a nice separation, but the majority appear to be in the benign category, which is what we'd expect. But there are a few, maybe 10, 12% of them that appear to be in the pathogenic strong category. Okay? So it looks like the data is breaking out as we would expect it. So then, okay, we have a functional result, but what does it really mean? Well, we have to calibrate it or standardize it to other standards rather than just the nonsense and synonymous. So what you're looking at here are 4 different collections of known standards. And if you just look at the top row, it's ClinVar. So we go into the ClinVar database, take out all the variants that are considered pathogenic, consensus pathogenic variants from the various companies who report their data. We also take the non-benigns in the same way from that data set. And then we look to see how they break out across the 7 categories, and you can see those across the top of the graphic. If I bring your attention to the very top row of pathogenics, 206 pathogenics, 199 or 97% of those end up in the pathogenic strong category, which looks very good. And over on the right-hand side, on the second row, 329 of the benigns are in the benign strong category. So there's your sensitivity and specificity in the last 2 columns for each of those 4 different groups of standards. And you can see we're well into the 9% for sensitivity and specificity for all of these control sets. So this is operating very well. But take-home message here is that it's not perfect. It's not 100%. There is some error in some of these results, and that's true of all what we call MAVE analyses, these large high-throughput screens. There will be error, but this is really highly accurate from what we can tell so far. So this is an attempt to basically say, okay, on the basis of those results, what do we know about variants that have been assigned to other groups? So the first column there is ClinVar and Enigma. Enigma is a consortium that's focused on classifying variants in BRCA1 and 2. These are the numbers of variants that fall into the categories that, these are known variants, known assigned variants by ClinVar and Enigma. And you can see how they break out there for the 2,881 in that first column. Then when we add our big functional study to that, what are the classifications once we've added our data to what they had? And you can see the numerical change. So if you look at the third column, you see that we originally had 1761 VUS, now there's only 267 left. Where do they all go? Well, they've been assigned to the benign and the pathogenic categories using this functional data. So there's a huge dramatic shift in VUS. We've eliminated probably 90% of the VUS under these criteria. On the bottom are just missenses alone, excluding the nonsenses and the splice variants and things like that. Very similar effects, 1623 with only 237 remaining as VUS. So really a dramatic effect on the VUS group. This is a Sankey plot which shows that same version. So the left-hand side is the functional results and all the little categories of functional results with these 7 probability models that I mentioned, or categories that I mentioned. And then on the right-hand side is how they all get classified using that ACMG classification model that I mentioned earlier. And you can see that they collapse into the various groups of pathogenicity. And really the take-home message here is, if you look down at the bottom, we have 326 likely pathogenic and 459 pathogenic. So how many are in, for instance, Enigma? If we look at Enigma and say, how many known pathogenic variants do we have in this region of BRCA2? The answer is 12. You have a list there of about 800. So there's a huge jump in the classification of these types of variants. And then the other take-home message is that variants of uncertain significance, we start with many thousands. We have 608 left in total from this region. And again, this is all possible nucleotide changes. So many of these variants have never been seen in humans, but they will be over time as we do more and more testing. All of these variants are going to show up at some point in time. So now we have this huge catalog of all these variants, all possibilities. And when you find a patient 5 years from now with a new variant, the result will be sitting in a catalog for you to use. So now let's take it to the next step is, okay, we have these classified, we have these assigned pathogenicity or benignity, as some people like the other side of it. But what about risks of cancer? Are these groups of pathogenic strong, pathogenic moderate and supporting, are they really conferring the elevated risks of breast cancer that we would expect to see, and ovarian cancer as well? And if you look at this busy table here, the top row are the pathogenic strongs that I mentioned, right? The most likely to be pathogenic out of our group of variants. And you see that they're giving us an odds ratio of 4.45, which is obviously elevated risk of breast cancer. The same at the bottom with ovarian, 7.91, a very elevated risk of ovarian cancer with these variants. The rest of the rows are actually standards. So if you look at the bottom of the breast cancer group, you see protein truncating. These are known protein truncating mutations. We know these are risk. they're giving you about 5.5 to 6. The nonsense variants in a big functional assay, same thing. The known pathogenic missenses from Enigma, same thing. So it's 5.56 for those known nasty mutations, but about 4.5 for the missenses that we're finding in this assay. That suggests that there are some variants in our group that probably are a little bit slightly lower risk, or that all missense mutations have a slightly attenuated risk. There have been publications suggesting that missenses don't have quite the same amount of impact on risk as truncating mutations. We still have to figure that out, but these data would potentially agree with that, and we'll see. More studies will resolve that. But take-home message again, the pathogenic strong variants that we're seeing in this assay, the thousands of them that we're looking at, they do appear to be high-risk variants as a group. And then this is an attempt to do a cumulative risk model for that. And so the top bar, it's hard to see these, but sorry, the text on this is not great. The very bottom of that is the light blue color. That's the population-based risk, approximately 12% lifetime risk of breast cancer. The very top of that is your known pathogenics and your P-strongs. So the P-strongs are sort of the red color. It looks black to me on this screen. But essentially, you can see that there is that elevated high risk. We're coming up to about 40 to 50% lifetime risk of breast cancer when you have one of these variants that we're calling pathogenic strong. So let's summarize what we've discussed today. We evaluated approximately 7,000 single nucleotide variants in the DNA binding domain of BRCA2. We missed exactly one variant in the entire study, so that was great. There's not a lot of dropout. Case-controlled studies, when we take the variants that we see here in our functionally pathogenic or benign categories, we clearly see these elevated risks of breast cancer and ovarian cancer associated with these variants. With a ClinGen model, so the ClinGen ACG model that I've mentioned for variant classification, we've classified 785 of those single nucleotide variants as pathogenic or likely pathogenic, which is really a huge number compared to what's known today. And then the same, just as importantly, into the benign category, we can take all these variants of uncertain significance and say they're likely benign or benign. That means that patient is off the hook, at least in terms of that variant and what that variant means for them and their family. So this is quite useful information as well. So the carriers of these variants, we've done the functional study, we've done the risk estimation, we've done the classification. It now looks like the carriers of these likely pathogenic and pathogenic variants may now graduate to enhanced mammography, better screening, enhanced MRI, and potentially PARP inhibitor treatment if they're diagnosed with cancer. The other point I want to make is that because this is a functional assay, because of the way we do this, the result applies to populations around the world. If a variant is pathogenic, it's pathogenic in blacks, whites, Asians, Hispanics. It doesn't matter where you're from. It's going to have the same effect. The risks may be slightly different between those populations because the risks in each of those populations move around a little bit. For instance, in African Americans, there's more triple negative disease, so we see maybe a higher risk for BRCA2 and BRCA1. But the take-home message is that all these variants apply to all populations. We don't have to do ethnicity and racial type studies or separate cell lines and things like that to figure this out. The last thing I'd want to point out is that there is a companion manuscript to this from a group at the NCI, from Shaimshan's group. They have very similar data to ours. We've aligned the two data sets, and we see 91% correlation across these thousands and thousands of variants. So the data looks very solid. It looks very consistent across two major studies. These studies, these papers will be reported in manuscripts quite soon. Okay, I'll stop there. Thank you.
So we're going to take the questions at the end, just once all the panelists have spoken. And thank you so much for that wonderful presentation. I just want to just call out to everyone who's here in the audience. We'd love you, if you have questions at the end, to come to the microphones. It's a little more interactive, but you can also do it through the app, and we'll also be getting online questions. So now let's move on to our next topic. And we're hitting a lot of different areas. I think all of these areas have particular interest to different points in the trajectory in which we might be seeing people who are presenting for risk assessment or for treatment related to their increased risk. So we're going to move on to a discussion on updates on risk prediction models. And I think we all recognize that we understand a fair amount about some of these high-risk genes, the risk prediction. We miss a lot of people who don't have a true genetic predisposition, but might have a predisposition due to a variety of other factors. And so Dr. Antonis Antoniou, who is professor of cancer risk prediction from the Center for Cancer Genetics Epidemiology in the Department of Public Health and Primary Care at the University of Cambridge, is going to give us an update on risk prediction models.
Thank you, Claudine, and thank you to the organizers for the invitation to talk about our work. So these are my disclosures. So what I'll be discussing is work that we do as part of the cancer risk program that aims to personalize cancer risks on the basis of the combined effects of the known cancer risk factors to facilitate cancer risk stratification and therefore enable us to target the available prevention and early detection approaches to those most likely to benefit. But before I do that, I would like to remind everyone of the different of the phases in the cancer risk modeling process until we actually implement such risks in routine clinical practice. So the process usually starts with exploring the need for the risk model, collating the necessary data to identify and characterize the different risk factors, synthesize these into multifactorial risk prediction models, then assessing those in prospective studies for clinical validity, developing the user-friendly software that clinicians can use in routine clinical practice and addressing requirements as far as regulations are concerned, and then assessing their clinical utility and doing studies to operationalize this in routine clinical practice. And within the cancer risk program, we address all these phases. So what I'll be discussing is some highlights of the work that we do in the different areas, starting with the development of the risk model. So cancer risk relies on the Bodicea multifactorial models for breast and ovarian cancer that consider the joint effects of rare genetic variants, polygenic scores, the effects of lifestyle, hormonal and active risk factors, mammographic density, cancer family history in an explicit form, tumor pathology in the case of breast cancer, which are combined then with cancer incidences to provide the individualized, the personalized multifactorial cancer risk. So looking at each of these in turn, this table summarizes the list of genes in which pathogenic variants are associated with breast and ovarian cancer in the model. So you can see we have 8 genes in the case of breast cancer, 6 in the case of ovarian cancer. There's a large overlap between the two, and which is one of the reasons that we model both diseases together. And this slide, which you've seen also from Fergus, summarizes the average risks that are assumed within the models for the breast cancer susceptibility genes. So we have the high-risk genes, BRCA1, BRCA2, and PALB2, and then the more moderate risk genes, which all have similar risks varying between 21 to 24% by age 18. So the models also include the polygenic scores. So there's a default polygenic score that is included in the model that was developed by the Breast Cancer Association Consortium based on 313 SNPs that explains around 20% of the familial risk of breast cancer. It was developed primarily using data on European ancestry women and which has been validated prospectively, again, in European ancestry cohorts, showing an association with breast cancer with odds ratio of around 1.61% deviation increase in the polygenic score. One of the questions when we come to model the joint effects of rare genetic variants and the polygenic scores is how the two interact, and there have been several studies now published in this area that have demonstrated that these polygenic scores also modify the cancer risks for rare pathogenic variant carriers as well. The data in the case of BRCA1 and BRCA2, but similar arguments apply to the other genes as well. One of the challenges when it comes to implementing these polygenic scores in practice is that based on the genotyping technologies that we use, we may not necessarily be able to capture all the SNPs that are included in the polygenic scores. Also, there are many different statistical methods that can be used to provide valid polygenic scores. So our models allow for alternative polygenic scores to be included in the model as long as we define the proportion of the familial polygenic component that is explained by the polygenic score, this proportion alpha here. And this ensures that there's no double counting of the effect of family history and the polygenic score, and ensuring that the models remain internally consistent, unlike some other models. So there's an easy way to calculate this proportion that we published last year, and it has been done now for a number of different polygenic scores that are used in clinical implementation studies, including, for example, the IMAGE consortium here in the US and the Wisdom trial. And this is the final component in the models, the lifestyle, hormonal, and reproductive factors. You can see the list of risk factors that are included here, including mammographic density in the case of breast cancer, coded using the BI-RADS classification system. Now, one of the challenges that we've had so far is that most of the data that we've had available have been on women who were from white populations or of European ancestry. So those models that have been developed until recently were more applicable to those populations. But we are now seeing more data emerging on more ethnically diverse populations as well. So a major focus of our work in the past year or so has been adapting these models to ethnically diverse populations. And despite the fact that countries can be used across different countries, again, one of the challenges here is that the ethnic makeup can vary substantially between countries. So what we have been doing is develop a general methodology that can be extended across different countries. But here I'm using the UK as an exemplar. This was our starting point. So we use two concepts here, ethnicity, which is self-reported, and which is more of a social construct in order to model the lifestyle and hormonal risk factors in the models, but also genetically determined ancestry to model the genetic components of the models. But the two are highly correlated. So looking at each of the components of the models, so we used data from the cancer registries in the UK to obtain the age-specific cancer incidences. So this is an example in the case of breast cancer, where you can see that the incidences are highest among whites, but the incidence of breast cancer is lower among Black or Asian women. So as I mentioned earlier, the models also include information on the distributions of different tumor subtypes. So we use data here from the National Cancer Registration dataset in England to obtain the age-specific distributions of the tumor subtypes in the different ethnic groups. And this is an example in the case of the proportion of estrogen receptor-positive tumors across the different groups, where, as you see, the majority of white women tend to develop estrogen receptor-positive breast cancer, whereas this proportion is much lower among Black women. Something that, of course, kind of is known, but this is now explicitly modeled in the in body CI and CAN risk. So there have been a few studies that have been published now that looked at the estimates of the relative risks for pathogenic variants across different populations. And the picture that is emerging from these studies is that, in fact, the relative risks appear to be very similar. This is the main assumption that we make in the models, that the relative risks remain constant across the different ethnic groups, but there are some differences when it comes to the pathogenic variant frequencies. But because despite the fact that the relative risks are similar, because the incidences are different across the different ethnic groups, the absolute risks of developing breast cancer will be different for carriers of pathogenic variants from different ethnic groups. So, for example, here in the case of CHEK2 carrier, if they are white, the risk will be predicted to be around 24% by age 80 compared to 17% for those of Asian ancestry. It is now well documented that the polygenic scores perform differently across populations, and this is the performance of the 313 SNP polygenic score, which was developed in European ancestry individuals, as I mentioned earlier, in the other ethnic groups. So what we are seeing is that these polygenic scores are in fact predictive of breast cancer risk, but they have attenuated effect sizes. Sorry, excuse me. So this difference is included in the models by re-estimating this alpha parameter that I mentioned earlier, which is the proportion of the familial polygenic component explained by the polygenic score. So as you can see, it's lowest among African ancestry women, which captures this kind of lower predictive ability in that group. And then we also take account the differences in the distributions of the polygenic scores across the different ancestries. These are data from UK Biobank, where, as you can see, the PRS mean is lowest among European ancestry women. And of course, this is used in the process of calculating the polygenic score input parameters. And I should say here that, of course, kind of as alternative polygenic scores are developed, which are ethnicity specific and or multi-ancestry polygenic scores, this can be easily be included in the models. And the final component here are the lifestyle and hormonal risk factors. And again, a systematic review that was published earlier this year has demonstrated, in fact, that the relative risks of the different lifestyle and hormonal risk factors are very similar across different ethnic groups, but what differs is the distribution of those risk factors, for example, BMI, parity. So here we use survey data from England to input the ethnicity-specific distributions. So having now this defined the model, this has implications for the risk stratification, and I'll try to demonstrate this here with a couple of examples. So here we are predicting the risk for the average woman in the population. So on the x-axis here you have the predicted risk by age 80, and the shaded regions here in this graph correspond to the 3 risk categories as defined by the NICE guidelines in the UK. So high risk, for example, are women who have risks of 30% or greater. So if we use just the lifestyle and hormonal risk factors to predict the risks, we get this distribution shown here in green, where, as you can see, only a small proportion of women will be identified as being in the moderate risk category. However, when we use the polygenic scores, we get a wider distribution shown here in yellow. So what this is saying is that the polygenic score is a better risk stratifier compared to the lifestyle and hormonal risk factors alone. But then we maximize risk stratification when we use the full multifactorial model shown here in blue, such that now around 18% of women will be classified into the moderate or high risk categories. So that was the model that is applicable in the general population. If we use now the ethnicity-specific model and we look at the risk distribution among whites, this is the distribution we get here in red, where around 20% will be falling into the moderate and high-risk categories. But when we look at distributions among Black and Asian women, as you can see, these distributions now are shifted towards having a lower risk, but also they are narrower. And this is because of 2 reasons. The lower cancer incidences in those groups, but also the narrower polygenic risk distributions. So in this case, if we were to use the existing risk thresholds as defined by the NICE guidelines to classify women into the different risk categories, we'll be identifying only around 5% of women falling into the moderate or high risk groups. Of course, kind of these are expected to change as we develop better ethnicity specific polygenic scores. We can do the same for carriers of pathogenic variants. An example in the case of CHEK2 variant, where on average they would fall into the moderate risk category with risks of around 22 to 23%. But then when we look at the full multifactorial model, as you can see, around 55% of the CHEK2 pathogenic variant carriers will be reclassified in either the population or higher categories with important implications for their clinical management. So the next step is assessing the clinical validity of these models in independent prospective studies. So there have been several studies now published looking into this. I'll describe the results from the 3 more recently published studies from different clinical settings. Starting with the latest study that is currently in press. This was in the UK Biobank of around 211,000 participants, where we had data on all the risk factors that are included in the model except mammographic density. So in these graphs, we see the calibration of the model, where you can see kind of the predicted risk on the x-axis, the observed risk on the y-axis, and then women are classified into deciles of predicted risk. So what we are seeing is that the model appears to be well calibrated in different risk categories, both for women under age 50 and women over age 50, with an area under the curve of around 66%. Another study that was published a couple of years ago was done now in the mammographic screening setting using the KARMA mammographic screening cohort that we heard earlier in the general session. So here we had data on all the different risk factors that are included in the model, including mammographic density. So the main result here, when we look at full multifactorial model, as you can see, the model remains well calibrated. The area under the curve here was around 70%. And if we take the 50% of the women with the highest assigned risks, those identified around 81% of the incident cancers that occurred in the observation period. So you target half of the population, but you identify the majority of the cancers. So these models used mammographic density coded as using the BATS classification system. More recently, we've extended these models to also include the continuous measures of density, the Volpara volumetric measure, Stratus software that was developed by the Karolinska group. So what we are seeing is, in fact, an improvement in the discriminative ability of these models using the continuous measure of density compared to using BI-RADS, and around 25% of women are reclassified into either a lower risk or higher risk category compared to using BI-RADS. And the final study that I'm showing here is in a prospective cohort of unaffected BRCA1 and BRCA2 mutation carriers, where again we see the model being well calibrated in quintiles of predicted risks and to discriminate well. And interestingly here, what we are seeing is that we can identify groups of mutation carriers who have low enough risks and who remain unaffected, for example, over the 5-year period, and which may be informative in the decision process, for example, in the short term for decisions around risk-reducing interventions. So the next step in the process is developing the user-friendly software. So in our case, we work closely with healthcare professionals to co-develop the CanRisk tools. They are available at this web address here, canrisk.org. They're freely available. These have gained regulatory approval for use as medical devices in the European Union and the UK, and they have been translated in 7 different languages. So the users can input the different risk factor information, and then the risks are produced in a number of different formats that to risk communication. And there is also a separate PDF that is report that is co-developed with patients and public and the healthcare professionals, which can be used to discuss kind of the information with the counselees. And this again is available in 7 different languages. It's an example here in Spanish. I should say that there's also a freely available API for third-party software to use the tool rather than putting the information interactively. And we have also more recently developed a new public-facing tool to address one of the challenges of having to enter this data during a consultation. We know that time is limited. So this enables patients to input their information directly, and then that's securely transferred to the healthcare professional, which auto-populates the CanRisk session. And this is currently being used in some of the implementation studies that we are doing. It's been evaluated there before general release. And which takes me to the last part on assessing the clinical utility and how we can operationalize such tools in clinical practice. I don't have much time here to get into individual study details, but other than say that there are multiple studies that are currently being done that try to investigate how we can provide multifactorial personalized cancer risks in different clinical settings, in clinical genetics, at screening, in primary care, including, for example, here in the US with the IMAGE consortium. And there is a study that is being presented in the symposium here that is done at the screening level in Denmark. We expect that these studies will be reporting in the near future. So that, I would like to finish by thanking all our study participants, our patient public partners, the teams at Cambridge and collaborators, and our funders. So thank you.
Okay, terrific. So for our final presentation, we're now going to move to the area of more therapeutics, where we have, I think many of us have started to use PARP inhibitors in the setting of treatment of our BRCA1, BRCA2 carriers, and sometimes for our PALB2 carriers as well. So Dr. Kate Nathanson is going to talk to us about reversion mutations in BRCA1 and 2 in response for therapy. And Dr. Nathanson is a professor of medicine and genetics. She's a deputy director of the Abramson Cancer Center and the Pearl Basser Professor for BRCA-related research at the University of Pennsylvania. Welcome.
I would like to thank the organizers for allowing me to present our data. I want to say that this is really a collaboration with Foundation Medicine, which is led by Brennan Becker. I really am honored and privileged to work with them and honored to present the data on their behalf as well today. Hopefully, I guess I will use this. Oh, I need to move there? Okay, all right, thank you. I have no financial disclosures. So just to remind everybody about our understanding currently about resistance to PARP inhibitors and platinum DNA damaging agents. Multiple mechanisms of resistance to PARP inhibitors have been described. On the top row, this is a very nice review from Jonkers showing drug or target-related resistance. On the lower row, showing mechanisms that have been associated with restoration of replication fork stability. Most of these mechanisms have resistance, have been really described in the laboratory or preclinical setting, whereas really the most common clinically described mechanism of resistance is restoration of homologous recombination, principally through what we call reversion mutations. So those are secondary mutations on top of the original etiology mutation in the DNA damage or homologous recombination gene that revert back to open reading frame and allow functional translation of the gene with usually, and we'll get into this in more detail, hypomorphic or decreased function as compared to the wild type. So PARP inhibitor resistance has been due to reversion mutations, was originally described in preclinical studies, actually, which described this with large genomic deletions. Reversion mutations have been described to date in 5 homologous recombination genes, BRCA1, BRCA2, PALB2, RAD51E, and RAD51D, and they're well documented to be associated with resistance to both PARP inhibition and platinum agents. However, the studies to date have had multiple limitations. They've been usually small, mainly single cancer studies focused predominantly on ovarian cancer. There have been a few larger studies, which have been compilations of prior data, not prospectively done across multiple tumor types. The bioinformatics technology to date has really focused and identified smaller reversions. There have been a limited number of progression biopsies, with many sampling only one site, and the genotype associations with resistance, with response to therapy have been inconsistent. So you want to know if you respond better or worse. For example, with a mutation in exon 11 with BRCA1 or 2, there is not really consistent data, and that might give you hints about reversion mutations. There was a very nice summary published this year from Colette, all sort of looking at what has been proposed about the landscape of reversions. They proposed several hotspots. Those are indicated by the specific mutations above BRCA1 and 2, and they propose specific reversion in different regions, some being single nucleotide variant reversions or just nucleotide reversions, with some having larger deletions. So the larger deletions being over exons 11 of BRCA1 and 2, and also 5' in BRCA2 with the larger deletions, and the smaller deletions in the DNA binding domain and the ring finger domain, as well as the BRCA-T domain. It's estimated that BRCA1 and 2 reversions account for about 10 to 40% of cases with PARP resistance. The reversion that emerges depends on the primary mutation, but the large deletions initially observed in the preclinical setting have not been reported as much to date, and it's been postulated that BRCA2 reversions may need an additional alteration to drive HR resistance restoration. I've got to do this carefully. Okay. However, there are many unanswered questions about homologous recombination reversions. Which genes have reversions? Are there differences in reversion frequency by gene, by tumor type? What's the frequency of reversions by pathogenic variant type? How do the reversions vary depending on where you are located in the gene? Are there hot, cold, and hot spots? And are there common recurrent mutations which have been of interest for targeting and therapy? The large Foundation medicine sample set was evaluated for reversions, as I mentioned. This originally encompassed almost 600,000 sequences that were done, and then samples with mutations in the genes in which reversions were observed. And you'll note that, and I'll talk about this later, there are 3 additional genes that were noted to have reversions, were narrowed down even further, and then samples with no evidence of a high tumor mutational burden state were narrowed down to about almost 39,000. And then of those, 717 samples were found to have reversions. It's important to note a few things about this. One, patients have multiple reversions. So 717 samples actually equals many, many more reversions than that. And 2, because it's a foundation or a testing medicine set, we don't know the status. Were they treated with PARP inhibitors? Were they not treated with PARP inhibitors, for example? So we don't have that data. So there are several mechanisms of reversion. Their reversions are the primary mutations are the nucleotide. You can have a substitution that reverts the primary mutation. You can have a deletion that encompasses the primary mutation, or you can have a secondary deletion or change that reverts several nucleotides down, a reversion mutation which restores the in frame. I'm terrible at this. There's also reversions at the exonic level. So one is at splice sites, and that we'll see that actually a very common mechanism of reversion in BRCA2, where you lose the exon, and then you go on to your reversion mutation. You can have homozygous copy loss of the exon, and then a reversion mutation within the exon. And then I'm going to tell you, these are actually, I think, most undetected and most complex types of reversions, which are around splice sites. So what can happen is that you can have reversion mutations that change the splice site, either opening up a cryptic splice site or mutating the certain splice site, and then you have novel splice sites. And these reversions can be very difficult to detect and aren't amenable to easily being found using common algorithms. So first of all, let's get to the data. Which genes are found to have reversions? So just to note that we identified 3 new genes with reversions. Not surprisingly, BARD1 and BRIP1 were newly identified as having reversion mutations, and CDK12 also has reversion mutations. Again, not so surprising. Important to note, in this large sample set, no reversion mutations were identified in other genes, including in ATM and CHEK2. And I think this really brings into question, I think some of us think about, is are the tumors in which these genes are drivers responsive to PARP or platinums in the same way? And I think this data contributes to that discussion. The other next, sort of looking at what's the baseline pathogenic variant type and origin. Just to note, they're generally mostly frame shifts, mostly nonsense. Interestingly, in WAD51C, you see a really enrichment of splice sites. And then these are the 3 novel genes with BRIP1 and BARD1 looking similar, CDK and CDK12 looking similar, looking slightly different with a large enrichment of rearrangements. Going along with that, when you look at whether or not the pathogenic origin, and this is estimated with Foundation's algorithms, was germline or somatic, not surprisingly, the CDK and CDK12 mutations are mostly somatic rather than being germline, whereas the others are reflective of a mostly germline origin. So what cancer types are these baseline pathogenic variants in reversion genes found? I just want to emphasize that on the y-axis is the percent of samples with pathogenic variants. Not surprisingly, ovarian cancer is by far the highest. Prostate coming next. You will see, though, differences in the frequency with prostate, for example, having a much higher rate of BRCA2 mutations and CDK12 variants, whereas, for example, breast, since that's the focus here, having a relatively equal amount of BRCA1 and 2 mutations. Just to note, oh, God, I'm not going to work on this. The reversions, these are the canonical tumors, and so this frequency of reversion mutations doesn't say anything about whether these are biallelic loss and whether these are drivers. This is just the frequency in baseline cancer genes. And I think it's really important to note that when you look at the overall rate of reversions by tumor type, so this means if you look on the y-axis, 4% of the tumors with these mutations have reversions, that then those other sort of tumors fall out, and really what you're seeing is reversions in canonical tumors. And what you see is clearly that the reversions are most common in BRCA2. And that's particularly noted in breast, but remember that ovarian is enriched for BRCA1, so this high level of BRCA2 in ovarian is relatively more common. You do have to consider sampling bias, I have to say, about why is the levels higher in breast and ovarian than pancreas and prostate, remembering that those can have more liquid biopsies, and we'll see on the next slide that they have more frequent reversion mutations identified than tumor biopsies. So are there differences in reversion detection by a biopsy type? I think not surprisingly to everyone that the reversion frequency in tumor biopsy or tumor sequencing is only about 2%, whereas in liquid biopsies in these tumors, it's somewhere around a little over 6%. That could be stage bias, definitely when these samples are being collected, but I think is not surprisingly, as you can sample multiple sites of tumors or probably multiple reversions at the same time. And although going along with that, when you have a tumor biopsy, you're much more likely to see 1 or 2 reversions, whereas when you have liquid biopsy, you see many more types of reversions. Then when we look at the reversion rate by the pathogenic variant prevalence, accounting for tumor gene and then tumor versus liquid biopsy, just to point out that the pathogenic variant frequency, not surprisingly, when you look at prostate and pancreatic cancers for PALB2 and BRCA1 is quite low. And then when you look at, gets higher for BRCA2, much for breast and ovarian cancer, and then not surprisingly, despite the relatively high prevalence of BRCA1 mutations in ovarian cancer, the rate of reversion relatively low. You then see in the tumor, when you look at liquid biopsy versus tumor, you see a much higher rate of reversion. See, it's almost 15% for the liquid biopsy in breast and ovarian cancer for BRCA2, whereas much lower tumor, only 5%. And then, interestingly, sort of trailing behind is BRCA1 ovarian cancer. And I think this is actually quite important when you think about this. So what is the rate of reversion by mutation type? It's important to note that despite this large sample set, no reversions were seen in patients with missense pathogenic variants, biallelic deletions, or rearrangements. There's only, if you look through literature and there's only one report of reversion by a missense pathogenic variant, and it's not clear if it's actually real when you read the paper closely. And so it's really important to think about that we are not seeing those reversions, probably because those are really hypomorphic and have some protein function. We see a low rate of reversions in splice sites. This is likely because, as I mentioned when I showed the types of reversions, you have to have very precise, very clear differences. It's not, you can't lead a large site, you have to have very precise changes to revert a splice site. And those may be harder and not as well selected for, harder to accomplish and not selective. Not surprisingly, frameshift having the highest and non having the next highest rate of reversions. This is a alluvial plot looking at what types of reversions that you see. Important to note, with the differences in DNA damage repair, differencing between BRCA1 and 2, we do see differences between the types of reversions and the types of mechanisms of the reversion mutations. So just to say, if you have large reversions or deletions greater than 10 amino acids, much more common in BRCA2 than it is in BRCA1. When you have frame restoration, interestingly, much more common in BRCA1 than in BRCA2. Missense variant reversions, much more common in BRCA1 than BRCA2. And interestingly, splice reversions, very common, you will see this in BRCA2, not seen in BRCA1. So the mechanism of correcting the reversion is, causing the reversion or becoming revertant is very different from BRCA1 to BRCA2. This is a complicated slide, so I'm going to walk you through it closely. So at the top, we have, I'm trying to see if, nope, you don't see it. At the top, we have the rates of reversion, so that's the black line. And then the red line is the density of the pathogenic variants. Then below that, this is for BRCA1, we have 198 pathogenic variants that are reverted, and then they have different types of reversions, and there's the pathogenic variants on the left, types of reversions you can see on the right, and then this is the plot showing the density and what types of pathogenic variants you have below. So you have the pathogenic variants above the lollipop plot, what's called a lollipop plot, and then the types of reversions are small reversions, less than 10 amino acids below. Then you see lines where you have more than 10 amino acids, and then you see the exonic deletions on the bottom. So let's go through this a little bit. So I think I'm going to hit with the highlights here. You see very few reversions before the reinitiation codon. So this is at the 185 del AG. As you note, there was reinitiation at 297. We don't really see any reversions. This is a known mechanism, by the way, a reinitiation of resistance. So not surprisingly, we see very few reversions over BRCA2-1. You can see there's a high pathogenic density and a relatively low density of reversions, and you can see very few in that region. There's some few exonic deletions. And then you can see in there are multiple reversions taking out part of exon 11. That's not surprising given the exon 11 splice variant is a known functional variant in BRCA1, and you can take a look at that and see that. I think what's interesting is we may be able to elucidate some biology. As you can see, there's actually all these reversions sort of stopping in the middle of this BRCA2-associated domain. What's happening there, what's that favored, I think is of interest. And then we can look here at what is the rate of reversions by mutations. Not surprisingly, the most common mutation that you see, and I shouldn't still call it 185 del AG, but 68, 69 del, has a very low rate of reversions, slightly higher. The next one would be because it's in exon 11. So then a similar plot for BRCA2 reversions, and again, I'm going to highlight some of the interesting findings here. Highest rate of reversions over the common founder, 59046 del, but it's not just at the founder, it's in that region, and high rate of reversions in exon 11, very low rate of reversions, and no reversions over 10 amino acids down at your helicoat domain and your OB1 domain, so very low rate of reversions in that particular region. And then extremely high rate of reversions that delete all or part of exon 11. It's important to note that not just you don't see the deletion of all of exon 11, you'll get loss of exon 11 by that huge splice site, which you can see right above there, and that actually means that exon 11 is lost. So there's loss of exon 11 here. We're also able to see some interesting biology. There's some very quiet regions in this particular, in exon 10. Obviously, very interesting to look at the biology here. Why is that happening? And just to note, not surprisingly, we see a relatively high rate of reversions, particularly for mutations that are common and found in exon 11. Do you have recurrent reversions? This is the founder. This is across many, 7 different individuals, and you can see that you have common reversions that happen to this same founder, and so we can see common reversions. This is just starting to map out PALB2 in the same way. The exon 4, where you see the large deletions, is in frame. Not surprisingly, we see in frame deletions, and we're starting to see lower rates of reversions above the WD40 as well. So does the size of deletions vary? Yes, not surprisingly, given what I've shown you to date, the size of deletions is highest in BRCA2 and highest in exon 11 of BRCA2. And it's important to note that 15% of all samples with a reversion in BRCA2 lose exon 11, and that exon deletions are frequent in both BRCA2 and BRCA1. So what can we infer functionally about the large exonic deletions? We did structural modeling. This is using AlphaFold from Greg Bowman's group. He's a structural modeler. And it's important to note that the functional domain structure of exons 15 through 25, which is what Fergus was taking a look at, remain largely intact with even the large exonic deletions. And in fact, they are almost 2 angstroms is considered extremely similar. And the deviation here is for exon 11 is just over 2 angstroms. And so the functional folding is all intact despite the lack of the large central exon. This is true. We did all of these for BRCA1. This is true as well for BRCA1. So I think finally, and I'm going to finish up, our non-revertant pathogenic variants associated with better outcomes. This is using the Foundation and the Flatiron data, and not surprisingly, when we put together the versions that are associated with with less types of reversions, they have longer outcomes. I think that's not a surprising but good to see outcome. So what have we learned? Genes in which reversions are observed include BRCA1, BRCA2, CDK12, PALB2, BARD1, BRIP1, RAD51C and D. Really brings up the question about pathogenic variants and other genes. We think that the contribution of reversions to PARP inhibitor resistance is likely underestimated. The loss of BRCA2 exon 11 really had not been previously elucidated, and with a complex spike reversions. I think this actually confirms the initial preclinical studies suggesting the importance of large deletions. And we know that the rates now vary by sample type, cancer type, pathogenic variant classification, gene, and position. I would not say that there are really specific hotspots as much as there are really specific low spots where we just see much lower rates of reversions depending on mutation type and location. And we think that reversions allow for hypomorphic function of these proteins. The lack of missense reversions allow use for retention of function, as well as the structural integrity for exons 15 and 11 for the lack of for the loss of exon 11. And it doesn't appear that we would suggest that the secondary alteration to restore HR function is not needed. And then just to note that BRCA2 reversions are much less common than in BRCA1 because there are multiple other mechanisms of resistance well elucidated by Neil Johnson and others that are associated with hypomorphic function. And that's it. Thank you. Sorry. I just want to acknowledge everybody and my...
Thank you so much. So the mic is now open for questions, but before we get started, I really want to turn this over to our patient advocate just to give some thoughts on this. All of this has applicability in the clinic, and I'd love to get Tanja Spanic, who's a PhD and a patient advocate for a long time. She's with Europa, Donna in Slovenia, and I'd just like to get your thoughts on some of these presentations to start with.
Thank you. Thank you, first of all, for the invitation, even though I have to say I have a PhD in neuroscience, not in breast cancer. So I happen to be a patient advocate because I was diagnosed with breast cancer 16 years ago. And I am a fan of genetics, so thank you for giving me the opportunity to be in this panel. But I would like to summarize or give a little bit of patient perspective to all of 3 talks. And I will start with the first one. Often we work as advocates and we meet individuals who get results of variants of uncertainty. This actually causes even more anxiety of unknown, what is going to happen, even if we get some kind of mutation or alteration that we know what does that mean. And actually your work brings a lot of hope for those individuals that we will soon have more clear answers. Which is essential to empower also patients to make informed decisions about the prevention and the treatment itself. So it's really crucial to think of new communication techniques and new ways how we can actually, as patient organizations, help build this bridge between the science and actually patients in the real life. And if I move to the second talk of Antoniou's, really impressive work, congratulations on this. And we believe that this risk prediction models are really invaluable for the community, for the population, and to identify individuals with a high risk, and also to bring diversity of the real world data, which what actually your model is also including. But in many countries around the globe, we see a lot of diversities that maybe some minority groups don't have access to all of this treatment. So this brings a lot of new work for advocates to advocate for access actually to all of these new tools, and to raise awareness, not just for the general public, but also for policymakers to make this kind of models available. And last talk, actually, I already see Andrew standing. Another disclosure, I'm also in the steering committee of Olympia trials. So yeah, I maybe see where this question is coming. But actually, yeah, impressive work of what you've been doing, and in this reversion variant, actually, this really brings more hope for patients that are actually not responding to the treatment anymore, and maybe also not just after they're not responding, but even before, how we will design the new treatments. And here I will stop and maybe add something more later.
Great, thank you so much. So why don't we go to some of the questions? So let's start off, Dr. Tudd.
Andrew Tudd, Institute of Cancer Research in King's College London. Thank you. Really, really fascinating session. I have two questions, if it's okay. One for Fergus Couch and for Kate Nathanson. So, Fergus, can I ask, I mean, at the moment, because many patients are now having sequencing results ordered by and delivered back to their oncologist, not to a cancer geneticist, through mainstreaming and other approaches. When that result comes back and it says it's a VUS, they're then not going to be suggested to have the therapy change that reclassification based on what you described to us might lead to happen. How do we resolve that? Is this through education? Is this now trying to get out there and say, you need to use this tool, or make that referral to your cancer genetics colleague who will be familiar with using this tool? Because this is going to have an impact on how people access proven therapies.
Yeah, it's a good question. There's no perfect solution to that, as you know. We will start by making the data available in as many different places as possible. As I mentioned earlier, catalogs of the variants and the results, put them up in as many websites, groups that can just advertise this information. In terms of getting it back to the patients, though, when they're not really going through genetics professionals, that's challenging. All we can do is try to educate our clinical colleagues in terms of the availability of the data and what it means. Certainly referrals to genetics always help in this situation. But you know, it comes down to intent of the physician and the time that they can put into it. So yeah, I'm not sure. I think maybe we have to do more broad outreach and education on the basis of these findings. And obviously it's not just BRCA2. BRCA1 has had this type of analysis done on it a number of years ago, and now there's other genes following shortly behind. Probably in the next 2 to 3 years, most of these key predisposition genes that we talked about will have been studied in this way. And so really the time is now, I think, to go to with educational programs to try and convince people to pay more attention to the sources of evidence and how they might use it.
I suppose as a proposal, just wonder whether the reporting companies who the oncologist is sending this to, when something comes back as, by their estimation of VUS, there's some prompt to perhaps discuss that VUS with a genetics professional, rather than it just be a reflex to VUS, you know, do nothing. That might help to push people to a deeper conversation, at least. And if I might ask a second question of Kate Nathanson. So Kate, you referred at the beginning of your talk to the fact that there were multiple reversions in some patients, and I know that a number of us have seen that. You also referred to the fact that there might be different levels of impact of those reversions leading to hypomorphic reverted alleles. Can you see in those that have multiple reversions levels of differences in the variant allele frequency that might inform on what reversions were fitter, or rather led the cell to be fitter than others? Does that help us understand the degree of hypomorphism, the sort of the relative frequency of each reversion type in the multiply reverted patient?
So that's a great question. I'm going to say two things, a few things about it. So one is some patients have up to 30 to 40 different reversions. So the numbers are really, really, really high. We have not gone back, and I think that's a great thing for us to do, to go back and look at what the relatively allele frequency is. But just to say, I think the other point that I was going to make during my talk is that one of the reasons that I think not seeing missense reversions is because we have so many instances where we see multiple different reversions. And so we would think if the missenses were reverting, that we would see something there. Do you see like a deletion or something, even if it just reverted back normal? I just forgot to make that point, and so I'm making that point, Andrew. But I think that's a great question. We can certainly take it back. I think it's hard, though, because it's a sampling issue, right? So where you do your biopsy, the biopsies tend to have one, but the liquid biopsies, it could be if you have a bigger chunk of tumor that has one reversion than another. You just what I'm trying to say. I think there might be sampling issues that may bias it, but I certainly can take it back, and we can certainly take a look.
Thank you.
No problem.
Kate, can I just ask you a question that came on online just to clarify for the audience? There were a couple of questions that asked about whether reversion mutations in these genes only occur in response or under the pressure of PARP inhibitor therapy or happen with tumor evolution, and how are they occurring?
Well, so that's a great question. I think that I would say this is a company data set. So it's very hard to know how people were treated, and because companies don't always get that information. Sometimes they do, and sometimes they don't. So the hypothesis has been obviously that it happens with therapy. And so this data set is not totally the right data set to look at because we just don't have all the information for that to answer that question. But I think it's a fantastic question and something that we can certainly see if they specify it's a primary tumor or something like that very specifically. But again, absence of information can be a problem here.
Terrific. Let me just go to that microphone over there.
Sure. Hi, can you hear me?
Yes.
Okay, Dr. Nicole Ramlachan from Trinidad and Tobago. From the clinical genetics side, just to go with the question earlier about the VUSs, when we receive the data from a genetic testing company and it comes back as a VUS, instantly when we give that to the referring oncologist or the patient, it's dismissed. I try my hardest as a geneticist to let them know that, well, this is not necessarily a negative, and we have to pay attention to it, especially when we see it occurring in multiple family members with varying types of cancer or similar progression of cancer. How do we approach that? Like I'm from the genetic side, the clinical genetic side, and how do we approach that in our reporting, and how do we the variation is there. And then also to my second question is, how does that VUS get changed from a VUS to a pathogenic mutation if it is you're seeing it in patients? But because we're from a very small demographic that's very racially diverse, we're not present in a lot of the big database populations that a lot of these tests are obviously done against. So that's my present dilemma.
I think in reality, this is going to fall on the testing companies. The expectation would be that they would send out, once they agree with the classifications, again, this was an academic lab that did this classification, we'll need to see expert panels and the companies themselves reclassifying the variants in the same way that we did. But once they've done that, we would hope that they would send out updated reports to the ordering the healthcare providers so that the information can get back to the patients. In the United States, I believe most of the companies do this already, probably on a 6-monthly basis. Right. They're sending out updated reports. Hopefully that would happen worldwide. And again, as I mentioned at the very end of my talk, the fact that you're in Trinidad doesn't matter, right? Because we'll have evaluated every possible variant in this region. And so it doesn't matter what population, the information should be available. And hopefully the company that did the testing for your patients will send out updated reports.
Okay, thanks.
Great. One last question, just in the interest of time, and then if others have questions, there are a few that are online. So, I didn't see way over there. Yeah. Okay, I'm sorry. You're suggesting maybe one more question over there. You were hitting the monitor for me.
Oh, okay, thank you. My name is Renisha Campbell. I'm a PhD student at UCSF, as well as a patient advocate with Tata Tuesdays, where we provide education to women newly diagnosed with breast cancer, but we're also very interested in prevention. So my question is in regards to the risk predictor. Has that been validated on individuals younger than 40? The traditional breast cancer risk calculators like the Gale model or the Cusack model, those are they're terrible when it comes to women, young women, as well as just women in general receiving a comprehensive breast cancer risk assessment. Most times that's not done. We've asked women, do you know your breast cancer risk? If it's low, moderate, or high, no one can ever answer that. They only know how to do self-breast exams. So I guess one is a statement in regards to there needs to be more individuals or physicians, healthcare providers doing comprehensive breast cancer risk assessments. And for the new one that's coming out, is that validated on young women?
Absolutely, this is a very good question. So the three studies that I've shown here, only the study in BRCA1 and BRCA2 mutation carriers included women younger than 40. The UK Biobank and also the Karma study all include women over 40. But there are other published studies in the UK Generation study, which has included women from age 20 onwards. So there are some validation data. I should say, without, unfortunately, mammographic density, though, kind of for those cohorts. And what we are lacking also now is we should be able to get additional data to validate these models in the ethnically diverse populations. This is currently lacking, so we really need the ethnically diverse prospective cohorts to be able to assess this.
Thank you.
I think in the interest of time, we're going to close. Thank you so much for the wonderful presentations, and thank you so much to the audience for your participation in this, and we hope that this was helpful to you. So have a good rest of your meeting.